everyone. My name is Masliana binti Zakaria. My student number is S52398. Welcome to AQU 4103 Ornamental Fish Culture class. So today, I will present to you about my lab practical video as I was assigned to culture one of the species of ornamental fish. And in this case, I choose mollyfish culture. This study is about to determine their true habitats, their behavior, as well as their larva rearing. So let's check it out for the task that I was assigned to.
There are two main varieties identified for mollyfish, which is sailfin and short-fin mollyfish. By the case, in this study, three common species are chosen, which is balloon mollyfish, common mollyfish, and Dalmatian mollyfish. All of these species was bought at aquatic store by the pair, which is 1:3 equal to 1 female to 3 males. After that, the glass aquarium are bought and check the leakage with 1/3 of water capacity. After few minutes, the cleaning process was began. The cleaning process were using brush and clean water. This is to remove unwanted objects that could affect the water quality and the fish as well. Next is gravel selection. The undergravel filter is a plate type of filter that is designed to pull water down through a media base which is known as substrate called gravel. This substrate is normally sit on the top of the plate, trapping the particles in the media as the water passes through it. These biological filters are usually run with acetone or a pump. If this filter is not maintained properly, it will contribute to high levels of nitrates as the media builds up an accumulation of detritus and other organic matters as well. How to maintain this filter? First of all, the media base needs to be thick enough to trap the particles, but not too so thick that it defects the filtering process. A layer of about 1.5 inches should be sufficient. Although, the media should not be too fine. If it is, it will clog the filter plate. A sufficient air or water flow are needed in all areas under this filter. That spot will allow bad bacteria to grow as bad bacteria grow in oxygen-depleted environments. In this study, the beach sand were collected and cleaned thoroughly with the chlorine water. The sand also soaked with the chlorine water overnight and dry under sunlight for 3 to 4 days to remove the organism residue that might be pathogens which could give harmful effect to the captured species. Then, we move to the gravel filter installation. The process starts from the gravel plate that was placed on the bottom of the tank, and then the tube as well as the acetone and aeration tube also was placed. Then, the gravel was placed on the top of gravel plate with 1.5 inches of thickness. A thick or thin layer of gravel will causing the water current moving slowly, and at the end there is lack of oxygen happened. The sand must be placed eventually. For the water filling, a plastic bag are needed which been installed at the bottom of the tank to ensure that the gravel wasn't moving when the water filling process. Meanwhile, a few drops of anti-chlorine are mixed together with the water. So basically, this is the correct water depth for undergravel filter system. The oxygen will push the water and form a circulating flow. Hence, air pump also needed to provide the oxygen. Plus, biofoam filter also used to support the oxygen. The water current were checked to make sure that the gravel filter worked well. For decorations, aquatic plants are chosen as the mollyfish require a lot of aquatic plant for spawning. These aquatic plants are bought from the online store and quarantined 1 to 3 days before it was placed in the aquarium to remove the fertilizer residue from the previous store. After that, the concept and technique of aquarium decoration. In this study, I used many types of aquatic plants that have their own benefits. The aquatic plant that I use in this study such as Java moss, streambok moss, waterweed and duckweed, which consist their own benefits as I mentioned in the slide. The submerged plants such as Java moss and Fluviatilis are tied together with something heavy like rock, driftwood and shell as well by using string. The outer part of plastic bag must be rinsed with the clean water. The water from management tank are introduced liter by liter into the acclimation tank by using siphoning technique. And then the bag was left in the acclimation tank for 30 minutes. The bag are then opened and the water were introduced to the fish by scooping the water liter by liter with hand. This is to allow the fish to adapt with the temperature in the management tank. The bag are left open until all the fish swim out. By using scope net, all the fish are transferred into the management tank one by one. This step is to make sure that the ammonia properties are not affect the fish new environment. Sometimes, the water from plastic bag can be used, but normally, the previous store have a bad quality of water system, which encourage the ammonia level to be increased. After 24 hours, the fish was fed with formulated pellet with a mix of spirulina and been fed for twice daily. Then, we move to the routine, which is water quality parameter check. As you can see, this is normal range for mollyfish water parameter. However, the parameters were tested once a week. The chemical characteristics such as ammonia, pH, dissolved oxygen, and temperature were tested, and the result is the DO of the water were 3.8 mg per liter, as the normal range should be above than 5 mg per liter. Due to this, all the fish are gradually died. Because all of the fish were died gradually, I bought another pair of the mollyfish and start to culture again. After a week since they were bought, the fish start to show an interest towards each other. This is the gender identification between female and male. The characteristic can be observed by using the points below. First of all, for the anal fin, the male have the anal fin that points backward, while the female have rounded anal fin. Next is for the color. Male have brighter and bolder color than female, and this can attract the female to spawn with the male. For the abdomen, the male consists of flat belly while the female have rounded and obvious pregnant spot. As you can see in the picture, the female has been pregnant for the few weeks after they were bought from the store. And then the size differentiation. The male are larger than female, as you can see in the picture. In this study, the fish start to show their spawning behavior. The male are seen to poke the female's belly, which shows that the males are interested to the females. Within few weeks, the fry are seen to be hiding in the accessories like as you can see. The fry is hiding in the driftwood from the fish as they always mistaken from the fry to the food. After the larva were transferred into the another tank, the larva was cultured and fed with a mesh pellet.